Hello, I'm Dr. Hope Rugo, your guest host of the ASCO Daily News Podcast today. I'm a professor of medicine and director of breast oncology and clinical trials education at the University of California, San Francisco's Comprehensive Cancer Center. Hormone receptor-positive breast cancer is the most common subset of this disease, and breast cancer is the most common cancer diagnosed in women worldwide. Endocrine therapy, or ET, is the cornerstone of management in hormone receptor-positive breast cancer and may involve suppressing estrogen production with aromatase inhibitors in the cancer cell itself, or directly blocking the estrogen receptor pathway through selective estrogen receptor modulators such as tamoxifen, or the injectable selective estrogen receptor degrader, fulvestrant. However, despite the availability of these therapies, the largest unmet need in treatment of HR-positive, HER2-negative disease lies after progression on endocrine therapy. On today's episode, we'll be discussing the emerging generation of endocrine therapies in breast cancer, some of which were designed to overcome common mechanisms of endocrine resistance and the challenges relating to optimal sequence of therapeutic options. Today, I'm delighted to welcome the world-renowned breast cancer researcher, Dr. Nancy Davidson, to the podcast. She is the professor and executive vice president for clinical affairs at the Fred Hutchinson Cancer Center and professor of medicine at the University of Washington, as well as past president of ASCO and AACR. You'll find our full disclosures in the transcript of this episode, and disclosures of all guests on the podcast can be found in our transcripts at asco.org/DNPod. Nancy, it's great to have you on the podcast today. Thanks for being here.
Thank you so much, Hope, for the opportunity.
You've done incredible work in the area of treating hormone receptor-positive disease, but also in understanding some of the agents and pathways that are most important to our understanding of how we treat and really think about developing new drugs for hormone receptor-positive breast cancer. The first generation of anti-estrogen drugs really included selective estrogen receptor modulators, but now we have a new generation of these anti-estrogen drugs. And there's also orally administered selective estrogen receptor degraders, so-called SERDs, that we hope someday will be an alternative, with already one approved, for the injectable SERD fulvestrant. But there's a whole additional class of drugs, complete estrogen receptor antagonists, referred to as SERANs, and proteolysis targeting chimerics, PROTACs. These agents are all at various stages of development in both early and metastatic settings and really represent, I think, a confusing array of different treatments that are being studied, some of which, of course, are approved. Nancy, it would be great if you could tell us some about these exciting new agents, starting maybe with our approved drugs and moving on to the agents that we're studying in clinical trials.
Hope, this is a terrific example of how basic science has really begun to inform clinical practice. For us as breast cancer practitioners, it was really easy for a long time because we only had tamoxifen. And then things like the aromatase inhibitors as a means of decreasing the estrogen ligand came into practice, as did fulvestrant, as you mentioned, a selective estrogen receptor degrader. Thanks, though, to a lot of understanding about estrogen receptor biology, and specifically the discovery about a decade ago of estrogen receptor mutations, something that we didn't think existed for a long time, but which we now know exists in maybe a third to a half of breast cancers after exposure to aromatase inhibitors. That's led to really an explosion, this alphabet soup of possibilities that you talked about. So SERMs, selective estrogen receptor modulators, tamoxifen is the classic one, right? Something that acts as an agonist and as an antagonist, depending on the tissue. The SERDs, the selective estrogen receptor degraders, lead to an estrogen receptor destruction. Fulvestrant is the classic example of this right now. But, we're very excited that a second one has been FDA approved. That's elacestrant. It was approved earlier this year. And it has specifically been developed to try to target those estrogen receptor mutant breast cancers because those are the ones that are particularly resistant. So that's the first of this new generation of SERDs that has come on the market. As you say, a key advantage there is that it's oral. unlike fulvestrant, which requires 2 monthly injections and is very, very inconvenient and uncomfortable for patients. Other SERDs are coming along. I think another one that is well along in clinical development is giredestrant, which is in the same family and which is being tested in clinical trials of the classic varieties. It's been tested in a window trial looking at its ability to downregulate Ki67. It's being tested against standard of care in metastatic breast cancer, first-line metastatic breast cancer. And there are some studies that are going to begin looking at it in the adjuvant setting as well. There are other members of this particular family, but I think that elacestrant is the one that we have at our fingertips right now. And giredestrant is one that's certainly coming along in clinical trials, and we should look forward to those results. For those who are interested in the giredestrant, the trials in question right now that are going on are Persevera, which is in stage 4 breast cancer, and lidera, which is in the adjuvant setting. Now, that's one new category, or it's an old category with a new twist. A second, these agents that are called complete estrogen receptor antagonists, CERANs, as you talked about them. The key here is that what they do, the estrogen receptor has a couple of domains, and in particular, it has 2 different activation domains, AF1 and AF2. And these CERANs are complete estrogen receptor antagonists, so they block both those domains. And the hope would be that they might end up being more effective than the other agents that we have available to us right now. So those are also in clinical trial at the present time, and we're waiting to see whether or not there's going to be any value in that particular area that's going to be able to go forward for us. Another area that I think we want to talk about is the PROTACs, as you mentioned. These are proteolysis targeting chimeric entities. So this is a kind of a complicated situation where you have a kind of bivalent situation where you have something that binds both to the estrogen receptor and then also to E3 ubiquitinase. So it leads to degradation again of the agents. That's another area for us to be watching. And then finally, the SERCAs, SERCA. So those are the selective estrogen receptor covalent antagonists. And what they do is they bind very specifically to a cysteine 530, that exists in the estrogen receptor, but not in other steroid receptors. So that's where the selective part of this comes along. Now, these are all in various stages of clinical investigation. Some of them are pretty early at this point, but I think the ones that are well established are elacestrant. Giredestrant is coming along, as we just talked about. Camizestrant is also coming along through the SERENA series of trials. And imlunestrant is also coming on through the EMBER series of trials. Hope I would also be remiss if I didn't go back to an old drug that's a new drug, and that's lasofoxifene, which is also in the SERM family. Those who have been in our field for a while will remember that lasofoxifene was originally tested in the prevention setting, where it seemed to have some activity, but it came back into our interest because it has really strong activity against estrogen receptor mutant breast cancer models in the laboratory. And as a consequence of that, it's coming back into the clinic through a series of trials that are called the ELAINE trials, where we're looking to see whether or not it might also be better than fulvestrant. It too is an oral agent, so that's a real plus for us. And the first set of ELAINE trials would suggest that there's some nice activity with it without a lot of toxicity. So lots going on in this field. I think for all of these things, obviously, we're also working very hard to think about the toxicities. All of these, as I recall, are oral agents. So that gets rid of one huge toxicity, which is you don't have the need for some sort of injection. But they all do have some side effects. Frequently interesting, they're like GI side effects, more fatigue. In a couple of cases, there might be some concern about cardiac arrhythmias. But I think GI turns out to be one of the most important things that we have seen in some of these trials. Generally very well tolerated, though. I think another important question, which I'm sure is in your head and is in mine, is that how are we going to integrate these into what we already have? So I think a lot of the work right now is looking at patients who have already received an aromatase inhibitor plus a CDK4/6 inhibitor and are now going on to one of these new agents. But you might wonder, if they turn out to be effective and well tolerated, whether they too should be perhaps combined with CDK4/6 inhibitors in place of the aromatase inhibitors or the fulvestrant that we use now. So I think that we can imagine that those clinical trials are either in progress in some cases or will be coming on as we try to think about how to integrate these new approaches into our existing standard of care, which is already quite complicated, right? We've gone far from tamoxifen, which was good for everybody, to now a really complex algorithm about how we think about hormone therapy, both in early breast cancer and in metastatic breast cancer.
Well, that was a fabulous discussion about the new agents and our existing agents, and both the, I think, exciting aspects as well as the challenges. One question that comes up, I think, a lot, and this is, of course, a huge question in many ways, but for ER positive metastatic breast cancer, where these drugs are first being tested, maybe we'll talk about that first. There's one trial at SERENA-6 looking at camizestrant in patients who have developed evidence of an ESR1 mutation in circulating tumor DNA, who are on first-line therapy with an AI and a CDK4/6 inhibitor. What do you think about that study, and where do you think that progress might go? Of course, that's based on data from another trial, which wasn't definitive, but it's sort of a suggestion, without studying the sequencing in detail, that maybe you would have improved progression-free survival with that approach.
Yeah, by which you mean the idea of monitoring pre-SER1 mutations in circulating DNA and then making therapy changes based on that? Is that what you're talking about?
PATA-1 trial, yes. And that led to this huge SERENA-6 trial, which of course is now accruing.
We would love that, right? I think that obviously breast cancer for many years has been interested in trying to have circulating markers that we could use to help to guide our therapy in a more meaningful way than we have in the past. And I think that the trial that you talked about is certainly one that's trying to see whether that's a possibility for us based on information like the PATA trial. I'm hopeful it's going to work out. I would say let's see what that looks like, whether or not it's going to be useful or not. It seems to me in some cases, some of these trials have not been quite what we'd hoped for, but I don't think we necessarily had the molecular techniques or the sensitivity, nor did we necessarily have other things to move to. Because, those two approaches require both a really good test, but also the ability to use the test to define a new therapy that's going to lead to improved patient outcomes. And I think that these are ingredients that we now have available to us at our fingertips more than we did, say, a decade or 2 decades ago. Exciting approach.
I do think there's a lot of challenges inherent in this process, but I think of, monitoring blood on a regular basis and following up and then randomizing based on results. But I think this is an incredibly important trial, as you have mentioned, to try and think about moving past using circulating tumor cells, which didn't work, to just change blindly, to the next therapy, but more have a rational reason to change to a drug that may be more effective before the disease itself progresses. And just for our listeners, the unique aspect of trials like this in SERENA-6 is that you change therapy before you have evidence of disease progression, but only this molecular evidence of a mutation that is associated with resistance to the therapy you're on. So it's a really important question. We'll see what happens. As you mentioned, sometimes we're not clever enough to really get around the fact that there are multiple mutations driving resistance in this setting. So we'll see how straightforward this is. The question also comes up, and I think that's a question with many of these trials now, is they're randomizing patients after a progression on an AI and CDK4/6 inhibitor to receive the novel endocrine therapy or fulvestrant. One of, I think, the concerns on treating oncologists is that then you're sort of eliminating the possibility of a targeted agent in that setting. And of course, we have new targeted agents we're studying as well, AKT inhibitors and new inhibitors of the PI3 kinase pathway. So the newer trials are now combining, as you mentioned. There's a lot of concerns about drug-drug interactions here and how you might really combine them. And then there are triplet studies looking at CDK4/6 inhibitors, oral SERDs primarily, and CDK4/6 inhibitors. What's your thought on these, and will they really help to move the needle forward?
I think that's a really an interesting question, Hope, is whether or not we're going to find that combination therapies from the get-go are the way to go, or whether we're going to end up having to use maybe more serial therapies. Because not only is it a question about whether or not you can tackle all of these different resistance mechanisms simultaneously, but I think the other question is, as you say, are there negative drug-drug interactions, and are there toxicities that are intolerable? Although these are targeted in all cases and they're relatively benign in terms of side effects from the breast cancer perspective, they're not devoid of toxicity. And so I think that's going to be another issue for us, is whether they're well tolerated during the time that patients are taking them. I guess the other thing I always think about, Hope, too, is that it's hard to know about value of cancer care. But, we are talking about agents that are not inexpensive. When you think about the financial toxicity in addition to the side effect toxicity that you and I just talked about, trying to think about what that value is going to look like is going to be very important for us. Also, as somebody who worked for a long time in the lab and in the clinic, there are an infinite number of combinations that you and I could think about that are rational. And so the question is, how are we going to pick the ones that are the most rational, if you will, the ones that seem to be the most promising, and take them forward into clinical trials? Because clinical trials, patients are our most precious resource. So we want to make sure that we are bringing forward only those things that really seem to have a very strong foundation and opportunity to improve outcomes over time. Tough question for us to try to think about, as you've talked about. The other thing is that these trials are not only for postmenopausal women, which are the majority of patients, but we also want to target them to premenopausal women. So in those women, we're also looking at using an LHRH agonist on top of this, right? Because many of these things are really, at least so far, designed for the postmenopausal state. So stay tuned. Lots of work going on. I think one of the interesting things will be making the leap from using these in metastatic disease at time of progression to taking them forward into the early breast cancer space. Several of the agents are now beginning to do that because of very strong preclinical and clinical data to date.
So we have new agents that we're studying in the metastatic setting, and we've seen a trend to move fairly rapidly from phase 1b trials directly into phase 3 trials, because as you pointed out so clearly, there are a number of drugs in this setting, and we don't really know not only which agent is better, but even what class of agents is better. So as we move more quickly from phase 1b to phase 3, the question comes up about how we're going to study these agents optimally in the early stage setting. I think we all thought that maybe changing based on ctDNA evidence of a mutation might be an approach, but that's complicated. A big question for you is whether or not you think that's ready for prime time as it's being marketed as such. And then the second question is really, are we better changing our whole approach up front in high-risk disease, or should we wait until after patients are exposed to their endocrine therapy and then switch as we go along? The EMBER-4 trial is looking at that switching approach.
Yeah, boy, I think that this is obviously the billion-dollar question, right, for many companies and for many of us as investigators. I don't know that I have a crystal ball into what the best approach might be. We know that already some of these have been abandoned. For example, amcetestrant. So I don't even have to learn how to say it because it's been abandoned. It did go to phase 3, as you pointed out, in the advanced setting, looking at it with palbo versus letrozole plus palbo, and it didn't really show a whole lot that the company wanted to pursue. So I think that sometimes these things are going to be abandoned based on the metastatic setting, which is a large trial. And that's a trial, obviously, where it might be a little bit easier to do some of the circulating markers, as you talked about, than maybe in the early stage breast cancer setting. I think probably these early stage trials are going to end up being big. They're going to have to be pretty clean. I'm guessing that most companies and most investigators will want to target them towards high risk individuals, as you talked about, for two reasons. One is that ethically, I think we feel more comfortable with that these are individuals where standard therapies are maybe not serving them as well as we would like, and where we have information to think that we can at least have equipoise about a new approach. And the second is that from a trial design point of view, the event rate is likely to be higher. And therefore, you might get answers earlier and with a smaller clinical trial. So that's where I suspect we're going to go. But it's a challenging question. And I think that many investigators and many companies are really trying to struggle with that right now, because we do have so many options. And we're not quite sure how to, first of all, develop these new drugs, but then really importantly, put them in the context of our existing drugs. And as you say, we're also simultaneously trying to develop superior molecular or circulating markers to guide us. So there's so many variables that are going on right now in this field that, from my point of view, it makes it really exciting, but it also makes it pretty complicated both for investigators and for pharma as we try to think about how to position these going forward. But, what a great problem to have, Hope, because remember when you and I started in breast cancer, pretty much all you needed to know was tamoxifen. And I think you and I also probably started at the tail end of the time when we were using androgens and progestins and, agents that now have basically completely fallen out of favor. Estrogen, yeah. And their side effect profiles, were not as good. So it's a good problem to have. a nice situation when science can inform our clinical investigation, our clinical practice.
Yes, I mean, I think it'll be fascinating to see where we end up at the end. But of course, the complexities of this include the fact that sometimes just the clinical trial design leads to a less than positive result because of the way the trial itself has been designed, which I think is the nice part about moving these earlier into treatment of high risk disease, but also brings up the question of all sorts of areas we don't not really placed to discuss today, like how the statistical design is made, et cetera, but can sometimes result in poor evaluation of excellent agents. One of the questions that comes up, and you brought this up earlier, which I think is really important, is that we are in the era of combining our endocrine agents with targeted agents, and oral agents, in a fascinating way, really bring up drug-drug interactions in a way that the injectable fulvestrant hasn't. And aromatase inhibitors were kind of quiet on the impacting metabolism, unlike tamoxifen. But we are seeing some drug-drug interactions. And, certainly the discussions about using these agents may include the question about whether or not you're having more diarrhea or nausea or fatigue, or one drug causes photopsia, flashing lights, things like that. Keratitis is becoming a new toxicity to follow. How are we going to figure out how to sequence these drugs? And are they only going to work better, all of them as a class, in patients with ESR1 mutations in their tumors?
I think we don't know the answer to the second question yet. That's something that really needs to be sorted out. Even if they do, that's still a really important subset of patients, assuming that we continue to start with the aromatase inhibitors, right? Because that's where those things really seem to emerge right now. I don't know how we're going to figure those things out. I guess that I'm hoping that, maybe a couple of agents in these classes will become kind of the lead agents. And so we'll be able to do this work with a handful of things as opposed to a whole array of things. But we'll see. I think that even within classes, obviously, these agents are going to be slightly different, probably. And so it may be that one member of a class may be slightly better maybe not from an efficacy point of view, but from a toxicity point of view. And we'll just have to see how it goes. I don't think I have any magic answers about that.
Yes, it'll be interesting to see whether or not the agents work in sequence too. Could you use a PROTAC after an oral SERD, for example, or a SERAN? That'll be fascinating to see.
I'm hoping that preclinical modeling may help with that a little bit, though, of course, we all know that there are plenty of things that do well in preclinical models, GEMs and rodent models and PDXs. And sometimes those things translate nicely into clinical practice, but sometimes they don't.
You mentioned, Nancy, that when we started out, that it was a fairly simple decision about what endocrine therapies, and we ran out very quickly. We're seeing some of those old classes come up with new agents, and you mentioned lasofoxifene, the oral SERM that seems to have some benefits and works in the later line setting, and also can be combined with a CDK4/6 inhibitor, has data with abemaciclib. There's also a new androgen receptor agonist, the enobosarm, that's being tested as well. Do you think that these older mechanisms have a future as well?
I do. I think that because we'll be able to understand the biology better than we did in the past, right? A lot of our hormonal therapy was pretty empiric several decades ago. But I think with better understanding of mechanisms and better understanding of what the patterns of resistance might be for a particular tumor, that we might be able to think about those things. You're right there. There's a whole parallel universe right now in androgen receptor targeted therapies that we're not talking about today, both in perhaps in hormone responsive breast cancers, but also in triple negative breast cancers and a certain subset. And so that's an area where we probably need to be watching what our prostate cancer colleagues are doing as they develop these agents and thinking about where they might mechanistically make sense to apply in the breast cancer field. It won't be the first time that we've received insights from them, because remember, prostate cancer people knew about androgen receptor mutations a really long time before we figured out that they existed in estrogen receptor as well. So there's a nice crosstalk, I think, between the prostate cancer field and the breast cancer field in that regard.
That's an excellent point. And we learn a lot from our colleagues studying other malignancies, and prostate cancer has been a big area there for us, and hopefully will help us with studying these agents, because there's different toxicity profiles, of course, as well. And then you mentioned the approval of elacestrant, the first oral SERD to have regulatory approval, and it's approved in patients who have ESR1 mutations in their tumors. We're also studying it in the Elevate trial in combination with all of the different targeted agents, the CDK4/6 inhibitors, mTOR inhibitor, everolimus, and the PI3 kinase inhibitor alpelisib, to really try and understand how these can be optimally combined. Would you check for ESR1 mutations at diagnosis of metastatic disease, or would you do this more after progression or start of progression of disease on an aromatase inhibitor?
I tend to be a conservative, Hope, and so unless there's a clinical trial that might be able to be considered, I would tend to check it only after progression on the aromatase inhibitor. But I think some people are earlier adopters, and I suppose it's possible that they might want to know from the get-go. Having said that, I do think elacestrant has an approval after aromatase inhibitor, as I recall. So presumably the patient would have to have that exposure before you would be able to act on the ESR1 mutation by administering the oral SERD. This is also an area that may well change with time, right? As we develop different agents, as the agents have different requirements or different indications, and as we perhaps have better tests, it may be things that we don't do routinely now will become very routine in the future. And perhaps in series, like you talked about, in a serial fashion.
Absolutely. I think that's a really important comment about how we're going to think about treating hormone receptor positive disease. And potentially, we have new chemo options, of course, not part of our discussion today, in antibody drug conjugates. So the future is certainly challenging in terms of understanding this appropriate sequencing. We need data, but it's exciting to have these options. As you pointed out, this is a great problem to have, too many drugs with efficacy, to try and understand how to use these in the most appropriate way. And as you also pointed out, the use in young women is particularly important. Whether or not you need to suppress the ovaries with all of these new agents is going to be important to understand as well, I think, as a next step. I don't know if you have specific thoughts on that area as well. I mean, that may reduce toxicity for younger women.
I guess our approach right now has been to suppress them, but I agree with you. You wonder, if you look at some of the mechanisms of action, whether that's really a requirement biologically and medically, or whether it's something that's kind of a leftover from the approval process, that for the drug approval, if these women were suppressed. I think we need to work that out, because that's another area where certainly women could have ovaries removed. That's pretty straightforward, although it requires a surgical procedure. But otherwise, use of LHRH agonists continues to be injections, right? And so if we're trying to maximize convenience and minimize toxicity for women with any stage of breast cancer, anything we can do, it seems to me, to eliminate an injection is going to be a good thing. So important for us to be able to work that out as well. Even though numerically it's a smaller number of women, it's obviously a very important population of women.
Last question for you. One question that comes up and sent to me and panels all the time is, if you have done your next generation sequencing and you see an ESR1 mutation and a PIK3CA mutation, what do you prioritize in terms of your choice of treatment? Because we have the choice of either using elacestrant or fulvestrant in combination with an agent targeting the PI3 kinase pathway.
I don't know that there's a right answer to that medically right now. So when I talk about it with patients, I suggest to them that it's probably not one or the other. It's kind of the question of which one to use first. So we talk a little bit about side effect profiles. We talk a little bit about patient preference. Neither of these drugs is devoid of problems, but sometimes patients have pretty strong feelings about which set of side effects seems the least unattractive to them, or the possibility of having those side effects. And I know that if one isn't well tolerated, you can swap over to the other.
Absolutely. I think that's a very important way to think about next therapies in our era of not really knowing what's right. And this idea of shared decision making is so incredibly important. Nancy, thank you so much for sharing your insights and knowledge with us today. I could talk to you for hours about this.
Hope, thank you so much for the opportunity. It is an exciting area, and I really enjoyed talking with you about what we know and the many things that we don't yet know, but we're working on.
Indeed, it's certainly an exciting time. Thank you to our listeners for joining us today. And thanks to the ASCO Daily News Podcast for highlighting this important area. You'll find a link to all of the studies discussed today in the transcript of this episode. Finally, if you value the insights that you hear on the ASCO Daily News Podcast, please take a moment to rate, review, and subscribe wherever you get your podcasts. Thanks so much.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.